Welcome to the IOE Rough Cut. I'm Bruce Maxwell, Director of the IOE here at MSU. Today we have Ryan Thum with us. Ryan's an assistant professor in the Plant Science and Plant Pathology Department. He's an ecological geneticist. That close enough?
Yeah.
Okay. He has degrees from Florida State University yesterday and then a PhD from Dartmouth. So you're going to talk today about aquatic species, weedy species, and very interesting subject and has interesting ecology. Take it away.
Great, thanks. Thank you. Thanks for being here. There's some unfamiliar faces and then a lot of familiar faces, so I appreciate it. This is a great seminar series. I've really enjoyed coming to this whenever I can since I've been here about 5 years now. And so I'm really happy to be able to come here and share with you some of my ideas. Hey, Robert, welcome to the show. So I'm going to talk to you about this idea of precision weed management, and I'll focus it on one species that we work on several species in the lab, but this is one species that we work on the most. It's certainly what we've done the most research on. It's an aquatic species. I know there's probably, aside from the people who are here from my lab, there's probably nobody who works on aquatic plant species here. So, you know, I do think it's relevant to other systems, but I'll stay in my wheelhouse, which is this particular species, Eurasian Watermilfoil. Maybe I'll give you just a couple minutes, or one slide at least, on just kind of my basic interests, just to give you kind of the basic science framework that I operate out of. So as Bruce mentioned, my field of study is basically ecological genetics. And ecological genetics, very briefly, is just the study of genetics in natural populations. And so it's separated from other fields of genetics in a couple of ways. They're non-model organisms, which creates a number of logistical challenges working on them. And then the focus is typically on phenotypic traits that are important to the study organism's fitness, so things that affect survival and reproduction. And that really separates it from you know, genetics that's based in sort of crop, animal or plant improvement. Okay, so just a little bit different. For me, working on invasive plants, the kinds of traits that I'm interested in are things like vegetative growth, and in particular, herbicide response. Those are two traits that affect what I would call some invasiveness, how they spread and establish, and how effective sort of prescriptive control methods are on them. I'm particularly interested in hybridization. I won't get into it explicitly in this talk, but a lot of my basic research is on the role that hybridization plays in plant adaptation, particularly for invasive plants. It's implicit in this talk, but I won't get into it. But anyways, if you're interested in sort of these basic kinds of things, you know, you can talk to me about them. Where my lab is heading right now in terms of basic research is this idea of genetic architecture, which is just a maybe fancy way of saying how many different genes and alleles are involved in determining a phenotype that we're interested in. And so some key fundamental questions in evolutionary genetics would be, are adaptive traits, adaptive phenotypes, the effect of mutations at single genes that have large effects, so in other words, very simple genetic control, or are they based on multiple genes, each of which has smaller numbers of effects? And then really important for this work, and I'll come back to this at the end, is this idea, well, when we see the same phenotypes in different genetic backgrounds, do they occur in the same way? Is it the same genes and alleles sort of building those traits, or are there different ways to sort of skin the proverbial cat? So you can imagine that you can get the same phenotype in two different genotypes because the same genes are working, or you could have different sets of genes that lead to the same phenotypes. So I'll come back to that at the end. So this is just, you know, this is sort of my basic research interest. I'm not going to really get into much of that directly today, because what I want to talk to you about today is this idea of utilizing genetic information in applications. And so in my case, it's, can we utilize genetic information to more precisely manage invasive plants? So is there information in DNA sequence data that are fairly easy to collect that we can use to actually make decisions, right? So I'll just make an analogy real quick, right? You know a lot about yourself, but if you swab your cheek and send it to 23andMe, you learn a whole lot more about yourself, because there's information in your genes that you don't directly see that we can use, that can be used to predict traits that you have some probability of having. Okay, and so that's kind of what I mean. Can we use these genes to predict things that we're interested in. And so I'll kind of break down, this thing doesn't show up on this thing, but so I'll kind of go through this figure, which is just kind of a conceptual model through the talk. Okay, so I'm not going to get into it right now, but you'll see those icons as we go through. And at the very least, if we can't use genetic information to predict things, then maybe we can at least use it to inform and evaluate how we make management decisions. Okay, so I'll tell you briefly what I mean by precision weed management. I just have sort of completely ripped this definition from precision agriculture, but I'm just focusing in specifically on the weed management piece of it. So in words, it's site-specific management of weeds based on the recognition that there's variation within places over time and among different places. I'm particularly interested in genetic variation. So variation can mean all different kinds of things. It can be variation about the sites, variation with managers or management tactics and strategies. I'm going to focus this talk in on my wheelhouse, which is genetic variation. These are the most common weed management systems, as I think this guy will probably point out. He was a little disappointed when he found out this was about aquatic plants, which is okay. So traditional row crop agriculture systems and lawn care, right, is where most weed management is done. But there's a surprising amount of weed management done outside of those systems as well. My focus is on aquatic systems, so I work on aquatic plants. So here's an image from the Jefferson Slough, which is a site we have a project on in Cardwell, Montana. But I think the ideas that I'll talk to you about today are really relevant as well to, in particular, other systems like rangelands and natural lands, so public lands. And I think these systems are more similar to one another with what I'm talking about than they are to this for several really important reasons. One is that in these systems, we're typically targeting one or two species that we want to selectively remove and maintain an otherwise diverse assemblage of plants. And that's very different from here. It's the opposite of here. There's a lot of limited control options in what I'll just sort of call these natural systems. And a lot of that comes from, these are multi-use systems, so there's a lot of regulatory permitting and public perception issues around management for these. And because of that, there's a lot more stakeholders at the table making decisions than there typically is in sort of commodities kind of based stuff, right? So those are the systems I'm particularly passionate about. I think we have to think about them a lot differently from a management perspective, and hopefully the ideas that I have are helpful in doing that. Okay, so the basic tool for weed management is this herbicide dose response curve. And so all it is we have a focal herbicide on the x-axis in different concentrations, and the y-axis is just some measurement of the effect of that herbicide at that concentration on the test subject. And so in this case, I just have on the y-axis the amount of growth in the presence of a particular concentration of an herbicide relative to an untreated control. So it's kind of like the per capita effect on growth of the herbicide. In aquatic weed science, and I would argue it's probably pretty similar in rangeland and natural land weed science, this curve is thought of as a species-specific trait. So in other words, if I want to know, I can go out to a local pond, collect Eurasian watermilfoil, do this experiment, and then say, okay, this is how Eurasian watermilfoil responds to this particular herbicide. Right? So it's thinking of it as a species-specific trait. But that doesn't necessarily have to be the way that it works, right? So it could be that different genotypes have very similar responses, but there's variation among them, right? Not all genotypes necessarily have to respond to the same herbicide concentration in the same way. So for me, I work on clonal plants. So if I were to show you, this is just hypothetical, but if I were to show you a graph like this, these might be just these different lines, maybe just different genotypes of the same plant, or if you're working on a sexual organism, they may be different genetic backgrounds, different genetic things. But the upshot is that there can be genetic variation. And even what looks like pretty subtle genetic variation, like in this graph, right, can have pretty important impacts on the efficiency at which we manage populations. So, for example, here are these cooler colored lines at 3 parts per billion, right? We're getting over 90% control. Whereas in the warmer colored lines, you would need about 5 parts per billion to get over 90% control. That's a big difference in the amount of herbicide that you're using, right? It's a 66% increase. Looks really similar, right, to us, but the actual amount that you would use to have the same amount of effect would be very different. You could over-apply to these guys, and you could under-apply to those guys. And as much as herbicides are expensive, right, that difference in herbicide might make might have a meaningful impact.
So that's parts per million in what you're spraying on.
Yeah, so it could be parts per billion or parts per million. I think I said some of the, yeah. So it could be measured in whatever, but yeah, how much you're actually the target dose that you're going for in a population. Does that answer your question? We can have more extreme genetic variation as well. So here again is that sort of the idyllic species dose response curve. We may have genotypes that exhibit high levels of resistance, right? And in those cases, we may need to use so much herbicide that it wouldn't be worth it because the non-target effects might be unacceptable, right? So permitters don't allow an endless amount of herbicide to be applied. There's ranges in which they'll let you apply them. And so if we were dealing with a population that had these genotypes, right, we might use something like 4 parts per billion or 4 parts per million of a focal herbicide and not have anything near what we would want to have. In that case, it's just a waste of time, it's a waste of money, and it's putting a whole bunch of herbicides in an environment that isn't going to do what we want it to do. So I use those two slides to say, in as much as we could understand how much genetic variation is out there for herbicide response and measure it and use it to inform the way we manage, that would be beneficial. That's my basic premise in this talk. To just make that a little bit more realistic, we also have to think of that in terms of a temporal and spatial dynamic framework. So I have just a little cartoon here to illustrate that. So imagine we have a population that has some genetic variation in it. So again, I work on plants that are clonal plants. So the different colored dots may be different genotypes, and individuals with the same color are just different ramets of the same genus or just genetically identical individuals. If you're working on sexual organisms, then these might just be alleles at a gene that are involved in herbicide response. So let's just imagine that these red dots are more resistant to a particular herbicide use pattern. If we apply that herbicide, right, it might look like it did a really good job, right, from an ecologist perspective, if we're just looking at the impact on the population size. Looks like it did a good job. But if we had sort of some X-ray goggles that could tell us whether or not different genotypes survived proportionally or disproportionately, we might find that this red genotype, right, had higher survivorship relative to the other genotypes. So it looks good, but in fact, there's been selection. Right? So there's non-random death in that example. Well, it worked well last year, so we do it again the next year, we do it again the next year, right? And eventually, what we would expect is that what we were getting out of those treatments wouldn't be what they initially were. And we might have a population that's dominated by these resistant genotypes or genotypes, right? And so this is why it's important to consider potential temporal dynamics within sites. And we can think about genetic variation within individual sites. And of course, if there's a whole bunch of something, it's available to colonize and spread to other places, in my case, lakes. So this is a shout out to Bruce here. Bruce and Fabian Menard wrote a really nice paper in a journal a couple of years ago, making the case that if we're going to do weed management, and we understand that evolutionary dynamics can be important to that, we need to consider those evolutionary dynamics when we're managing weeds. And so 2 points that they make in that paper that will I think are really relevant to the stuff that I'll be showing, the empirical stuff I'll show you today, is this: evolution occurs in contemporary time, right? And it's just sort of a verifiable fact in most cases that there's genetic variation within and among sites, right? And so there's sort of fuel out there for evolutionary change. And then herbicide resistance isn't just confined to one area. It can sort of be linked spatially and temporally to other areas. And so in as much as that's true, then we would want to coordinate local and regional information and local and regional actions, management actions. So with that, then with all that background, I'll actually show you some data that I think are relevant to all of this. So the plant I'll be talking about is Eurasian Watermilfoil. It's this beautiful feathery leaved aquatic plant. And if you grow it in a clean aquarium, it's really, come by the lab, I'll show you. It's a beautiful plant. It's often sort of perceived more at this spatial scale, and it often sort of forms these really dense monocultures that are annoying to people. It interferes with recreation, navigation, irrigation, has pretty big impact on property values in lake-rich areas like the Great Lakes Basin. It's the most widely managed invasive aquatic plant in the northern tier of the US, and that's sort of how I ended up working on this species. It's just sort of the most relevant one in the areas that I've lived and done my work as an assistant professor. Most management is chemical or herbicidal, and within a state, they're regulated at the state level primarily, and within a state, there's pretty much universal prescription. So if a state allows the use of 2,4-D, for example, they're pretty prescriptive about how you can use it. And so in the way things are managed currently, there's very little, there's zero practice in terms of thinking about, well, maybe we want to use it this way in this site and this way in another site. They're pretty universal prescription. In terms of the biology that's important for this talk, it's a really extensive asexual reproducer, so it fragments very easily. Within a given lake, a lot of the biomass will have come from asexual reproduction. There's also quite a bit of genetic variation. These plants do sexually reproduce as well. So the first question, when I started working on this was, well, is there any genetic variation, right? If we have this idea that genetic variation might be important, it's probably pretty critical that you ask yourself whether or not there's any genetic variation in the first place. So this figure is just to illustrate that there's quite a bit of genetic variation. It's a principal coordinates analysis or an ordination of a couple hundred different molecular markers that are pretty easy for us to amplify. Every dot is an individual plant. The plants come from all over the country. Dots that are closer together are more genetically similar, and dots that are further apart are more genetically different from one. This cluster of plants over here is a native species, northern watermilfoil. It's the native sister species to Eurasian watermilfoil. They're pretty similar morphologically and ecologically. Then everything else, all of this stuff over here is what we would call Eurasian watermilfoil in a broad sense. And what that actually is Eurasian watermilfoil in the strict sense, and then Eurasian watermilfoil commonly hybridizes with northern watermilfoil, right? So this whole swath of genetic variation in here is hybrid genotypes between Eurasian and northern watermilfoil. And so a lot of the genetic diversity out there in what we call Eurasian watermilfoil comes from this hybridization between these two species. And that's a whole area that I'm really fascinated by and interested in, but I won't get into those details. Just suffice it to say that a lot of the genetic variation I'll be talking about comes from that sort of swath of genotypes. Okay, so there's a lot of genetic variation. All right, well, who cares? If it's not relevant to anything that a manager would be interested in, then it doesn't matter, right? There's a lot of genetic variation in this room. Presumably there are no identical twins in here, so everybody's genetically different. But in a lot of ways, we're very, very similar to one another, right? So the more important question, this tells us that there's the potential for there to be important genetic variation, but it doesn't prove that there is. So these next couple of slides, I'll show you data that just demonstrate that yes, there's genetic variation that managers should actually care about. So this was a study done a few years back. We just sort of haphazardly sampled 10 different hybrid genotypes that came from Michigan, Wisconsin, and Washington State. And so there's 3 panels here and a set of bars for each, and the bars correspond to each other. So the first bar here is the same genotype as the first bar there, and is the same genotype as the first bar there. These 2 red bars are 2 different Eurasian watermilfoil genotypes that we had in the lab that we knew were fast growers and we knew were sensitive to herbicides that we were interested in working with. And so they're there as references. And so I think what you can see is there's a lot of variation in growth. Oh, so sorry, the y-axis is just how much they grew from a standard length over a standard period of time. So just a measure of how fast they sort of intrinsically grow. This first panel is without any herbicide. These next 2 panels are 2 different concentrations of 2,4-D, which is a really commonly used herbicide on these plants. So we can statistically demonstrate that at least some fraction of the variation in growth rate in the presence and absence of this herbicide is due to genetic differences among individuals. And so these values are just called broad sense heritability values, and they basically just tell us what fraction of the overall variation in growth rate is due to differences between genotypes. And what's different between genotypes is the genes that they carry. So anywhere between 1/4 and 1/3 of all the phenotypic variation is a result of genetic differences between plants. So we don't know what genes are responsible for these differences, but we do know that it is genes that is responsible for those differences. This is a different way of looking at those same data. So I would argue this is the way that like sort of managers see plants. They see how much they grow and how much of them is out there. This is the way scientists think about this kind of stuff, I think, more likely, which is sort of the per capita effect of adding 2,4-D. So these are the same data, but for these two treatments, it's just how much the plants grew relative to an untreated control. And so this would be sort of more of a typical dose response curve. These red lines are the wild types that are referenced Eurasian Watermilfoil. You see there's a lot of variation in the proportional effects of 2,4-D on these genotypes as well. So either way you look at it, I would argue that whether you look at it the way I think a manager would see it, or if you look at it the way a scientist would see it, what these data tell us is that different amounts of 2,4-D are required to have the same impacts on different genotypes. Okay, so this idea of a universal prescription, hey, we use 2,4-D at this rate all the time, isn't really, in my opinion, such a good idea, because these data tell us that different amounts would be required to have the same impact. This line here is just for reference. So these are our fast-growing, untreated Eurasian Watermilfoil plants. And so even when we add 2,4-D, there are genotypes that are growing as fast as an untreated, fast-growing plant. So 2,4-D is basically off the table. If only we knew it, 2,4-D should be off the table for genotypes that have means that are above that red line. That's my opinion. That does not reflect the opinion of Montana State University. So then here's another example with a different herbicide. So fluoridone's another very commonly used herbicide to control Eurasian Watermilfoil. Several years back, we did a field study where we quantified the abundance of watermilfoil before treatment and after treatment with fluoridone. That's a very standard thing to do. We also included some measures of genetic variation. So fluoridone is typically very effective. What this figure shows is this is the outline of the lake, and then each point on that figure is a spot where there was watermilfoil present before the treatment. The white dots are spots where the watermilfoil went away after treatment. The gray dots are where abundance stayed relatively the same, and black dots are where abundance actually increased. And so typically what we see with a fluoridone treatment is we would see all white dots in here. And so this is clearly a failed fluoridone treatment, because we should see basically no milfoil in the lake, and we still have a ton of milfoil in the lake. This figure is kind of boring and it's kind of ridiculous, but this just shows genetic variation. So there's all one genotype present in the lake before and after, right? So that gives us a very strong indication that the reason we see variation in site to site is not like anything hydrological or chemical. It's because plants were actually surviving treatment. So, right, we collect those plants, we bring them into the lab and test this idea, whether or not they're actually resistant to fluoridone. This is a dose response curve for two different genotypes. This is a wild type genotype that reference we knew was susceptible to fluoridone. Fluoridone's used at 6 parts per billion in Michigan because of these data, right? 6 parts per billion, you don't expect there to be any milfoil growing, right? So that's the concentration that you're allowed to use fluoridone in Michigan. This is the genotype collected from that lake, and at 6 parts per billion fluoridone, it doesn't even know there's any fluoridone in the water. So fluoridone has basically zero effect on this plant. In other words, had we not treated with fluoridone and come back and we probably would have seen this exact same thing. So again, because this is done in the laboratory in a common garden, we know that the difference in the dose response curves between these two genotypes is a result of the genes that they carry and not anything else. So there's, again, genetically meaningful variation. This is just a nice illustration. This is from a colleague of mine, and really demonstrates the point that, well, anyway. So what they did was they sampled these 4 different genotypes from these different lakes, and then they exposed those genotypes to 3 different commonly used herbicides. This is all done in the lab. And these values in here are just the proportional growth relative to untreated controls. And the different columns are just different exposure times. We'll really deal with that. So if we look at this particular herbicide diclot, we have a genotype here, down in the bottom in the green, that's relatively sensitive to it, right? Not a lot of growth when you expose it to it, and then a genotype that's relatively resistant, right? So having proportionally more growth relative to that other genotype. If you look at those same two genotypes for this other herbicide, 2,4-D, right? Now the pattern is flipped, so this one is susceptible to 2,4-D, and the other one is relatively resistant to it. So if we had this information, right, at our fingertips when we are making management decisions, we could say, well, we have a range of options. Which is the best option? And for this genotype, we would probably choose 2,4-D, right? And choosing diclot would be not a wise decision if you had that information going into it. And for this other genotype, we'd probably choose diclot over 2,4-D, right? Pretty straightforward. And so one important point is that even if a genotype is resistant to one herbicide, it's not likely to be resistant to all possible options. So there's options in front of us if only we had that information available to us. So here's the basic idea. We have a lot of methods for doing aquatic vegetation surveys and evaluating efficacy at the whole population level. I would argue that if we can incorporate DNA information, like DNA fingerprinting stuff that we do, this is a little bit more, that in as much as we could have a library of genotypes that we have categorized, we could use that information to make permitting and control decisions. So that's the big idea, all right? It's pretty simple, but that's as complicated as I'm capable of. So I'll show you an example, an empirical example of where I think that sort of shows some promise for this idea. And then I'll poke holes in it after that. So we have a large project going in Michigan right now. And last year we surveyed 52 lakes across the state. And we just sampled a bunch of weeds and we genotyped. These data are from a genotyping method that we're developing with a graduate student of mine who's actually over here in the front row. And so these are based on a single nucleotide polymorphism. I think there was 500 or so that went into this. This is a really strong, robust data set. And this is a principal coordinates analysis, again, just like I showed you one of the first slides. So you just see these different clusters. So all just means that we have a lot of power to distinguish different genotypes with these markers. That's all that I want you to see from that. If we look at this particular genotype, and I've cherry picked this one, but it's kind of cool. We just look at this genotype. We found it in all of these lakes. We found it in 7 lakes total. I think there's 6 dots on there, so I'm missing one of them. But so out of 52 lakes, right, we found that genotype in more than 10% of the lakes that we surveyed. So it's a fairly common genotype. And what I mean by genotype is just they all have the same DNA fingerprint as well. And that suggests strongly that they're all the exact same plant, that they would have the same properties, right? They're clones of one another. It just so turns out that that genotype is this fluridone-resistant genotype. That's why I cherry-picked it, okay? So what I would argue is for those 7 lakes, right, there would not really be any compelling evidence to justify using fluridone in those lakes, right? I mean, a permitter, if they knew this, should probably say, probably not a good idea. Unless you can prove to me that it's going to work, probably not a really good idea. And in the absence of that information, what I'll tell you is every single one of those lakes will be treated with fluridone sometime in the next 5 years, because they get treated with fluridone about every 3 to 5 years. So, and fluridone is really expensive. And so we stand to lose, this information stands to be pretty important for those lakes. So that's the promise of this, right? It'd be really cool. If we could do that with every single genotype and every single lake, that would be great. The problem with it is that there's a lot of genetic variation out there. And there's too many genotypes to exhaustively characterize them all. So that's the real roadblock in front of this idea. What I'm gonna do for the rest of the talk is talk to you about how to get over those roadblocks in the short term versus in the long term. So in the short term, I'll show you data for this. I think it's just as simple as saying, let's build a library, but let's just do it in a judicious way. Let's just prioritize how we build the library. And I'll walk you through some examples of how to do that. And then in the long term, It's more the sort of 23andMe approach, right? If we can identify the genes and alleles that actually determine herbicide response, then we don't have to exhaustively characterize every unique genotype that we find. We can just do molecular genetic surveys and use those to predict the phenotypes that those things would have. And that's a really long-term, again, these are non-model organisms, they're plants, so they're a pain in the butt, things like polyploidy. So this is a real long-term vision. What I mean by short-term is like 5 to 10 years, and long-term is probably more like 10 and beyond. But in the meantime, people are going to manage anyway, so we might as well do something, right? Okay, so how do we go about prioritizing genotypes in the short term? Well, one way to do that is to just follow, like to incorporate genetic data into our aquatic vegetation monitoring. And as much as we see genotypes that show signs of increasing their relative frequencies after treatment compared to before treatment, that would be a big red flag to say that genotype might be one that we want to worry about. I'll show you some data for that. So this is St. Albans Bay. This is anybody from Minnesota in here? Chances are there's got to be one person from Minnesota in here.
Went to college there.
Oh, there you go. That's close enough. We'll take it. So this is St. Albans Bay. This is a bay in Lake Minnetonka, which is a very large lake outside of the Twin Cities area. And it's big. If you've ever seen it, it's just a whole bunch of lakes stuck together, basically. So this is a large bay in Lake Minnetonka. The points on here are areas where water milfoil was found before treatment. The gray polygons are areas that were targeted with treatment with the toxinic herbicide in early June in 2015. And the different colors and symbols are different genotypes of Eurasian watermilfoil that we distinguish with our molecular markers. And so this blue genotype was the most common, and there was a variety of less common genotypes. Herbicide treatment was really successful. Go back in September, and there's hardly any plants in the lake at all. We found 3 Eurasian watermilfoil plants, but all 3 of them were the same genotype, this yellow genotype, and that was relatively rare before treatment, and it's the only thing left over. So treatment was really good, right? But what's left over looks like it's maybe a non-random set of what was there. Or it could just be chance. So wait, there's more. So here's another bay on Lake Minnetonka treated in the same way at the same time. And so again, the different colors and symbols are just different genotypes. That yellow genotype is the same genotype from the one before. It was relatively more common in this bay than in the last bay. Overall treatment success was not nearly as good in North Arm Bay, and there would be a variety of explanations for that from an herbicide applicator's perspective, and they may very well be true, but we also see this yellow genotype, again, increasing in relative frequency post-treatment, in relative frequency, post-treatment compared to pre-treatment. So in 2 different bays, we're seeing the same genotype increase in relative frequency. So in my mind, right, that would be a sign to say, okay, that's a genotype that we might be interested in actually having some herbicide data on. What I'm not showing here is a whole bunch of bays in Lake Minnetonka, and this genotype is in all of them, right? So it's all over the place. So it's not like, you know, if we happen to get rid of it here, it's gone from everywhere. So there's a lot of impetus for understanding this particular genotype. So that's one way we could prioritize genotypes. Another way is just to sort of, you know, if we had a database of genotype occurrences, just sort them and rank them and say, well, the ones that are the most common and widespread and abundant are the ones that we should focus the most on. So we have another project in Minnesota where we surveyed like about 75 lakes or so. There's about 30 points on this graph or figure, and those are 30 lakes that all share the same genotype in Minnesota. And so all else being equal, if we had a precise understanding of how that particular genotype responded to herbicides, right, we could make the best sort of what has the most selectivity, what has the most efficacy, and what's the most cost effective. And if you integrate that over 31 different lakes, right, then the total amount of increasing efficiency is pretty high. So focusing on those genotypes would be good. And then these 2 are just showing you, in our survey in Michigan, we found a collection of lakes that shared genotypes with one another. So we could just use basic genetic survey information to give us some way of prioritizing genotypes. So if we do this, this is the way that this would work, right? You just incorporate genetic information into your traditional aquatic vegetation survey and monitoring. When you get lucky and you have a lake that has a genotype or a set of genotypes that have been characterized, you can use that information. And when you're unlucky and you don't have any information on it, you could ask yourself some questions to see whether or not you wanted to prioritize any of those genotypes. And then those would get added to the library. And then when we see them in other places or at other times, we would have information about them. So that's a short-term thing. There's a lot of interest in doing this from several different states. And so I thought I'd just spend just a few minutes to sort of lay out like the pieces that we're dealing with if we were going to turn that into some sort of service, for lack of a better way to put it. And I put these up here in case there's people in the room that have expertise in some of these areas or interest, then that might be avenues for us to interact. We need a sort of standard sampling protocol. I think we have a pretty decent handle on that right now. We're working on a genotyping method that would be more repeatable across labs. The kinds of things that we've done in the past are, they're rigorous, but they're hard to compare across labs. And so Jeff and I are working on a genotype method that would be more repeatable. The idea would be that, it's great that my lab does it, but it'd be a lot more efficient if other people could also contribute to that library. A really important statistical one is methods for detecting non-random change in genotype distribution. That's not that easy to do. We have some methods, but the power is really low, and I'm not a statistician, so I would certainly look for people to maybe be interested in that. I'll skip over this one for now. Rapid and effective growth and herbicide assays. I mean, they take a lot of time and take up a lot of space, so we're trying to figure out how we can do that more efficiently. At some rate, we want to validate what we see in the lab and make sure that it actually translates into the field. And we've done that in a couple of cases, and we're just got to kind of keep doing that. And then these ones are more, so this algorithm for prioritizing genotypes and centralized database and coordination. That's kind of database stuff. That's not also my area of expertise. And there's a lot of interest in sort of figuring out the best way to sort of put this information together, how to disseminate it. There's sort of social, political questions about who should be responsible. Should it be private and proprietary, or should it be publicly available? That kind of stuff. So those are things that with a variety of stakeholders in Michigan, Wisconsin, and Minnesota, we're having a lot of discussions about that stuff. I'm getting low on time, but let me just spend a few minutes getting to sort of the longer term picture for this. So the short term stuff is what we're doing now, and I think we'll continue to build that library. We're also chipping away at long term stuff. What I'm going to show you is just sort of a workflow to how to get to the sort of 23andMe. Hey, you send your DNA and we tell you everything you want to ever know about the plant. The good news is that everything that we're doing in the short term has to be done for the long-term studies anyways. So, it's not like they're independent lines of research. What we do now to help the short-term vision is helping the long-term vision. But when we find genotypes that differ in how they respond to herbicides, there's a variety of genetic methods that have been used for a very long time to identify genes or at least chromosomal segments that are associated with those traits. And so you can cross susceptible and resistant genotypes. You get a whole slew of progeny from them. And if you assay those progeny for their responses to herbicides, then you can tease apart. You can use some genetic and genomic techniques to tease apart the genes, or at least the chromosomal segments that are associated with those traits. And so these are folks that are in the room, PhD students that are working on this stuff. Jeff is working on quantitative trait locus mapping. These are not his data. I totally porked these from Google. And Greg is working on sort of gene expression differences related to herbicide resistance. And these are also not his data. I worked that from Google too. So if any of those figures are yours, I apologize, but I'm just doing the best I can. And then when you find those genes, and as much as those studies bear fruit and tell us what genes are associated, right, then it's quite simple to just assay those genes. So this to me is pretty overwhelming. It's a lot of work, and there's a lot of stress and anxiety about it. There's a lot of questions that keep me up at night related to those basic research interests that I talked about at the beginning. That could have a huge impact on the feasibility of having this kind of 23andMe approach. But I'm going to put all of that aside for a moment. So that's sort of the research program. And I'm going to leave you with just an example that, again, sort of shows you the promise of being able to do this. So it's like a lot like a cooking show. Like I'm just going to go right to the end and show you the beautiful cake, even though I'm not going to show you all the screw-ups that went into it. So this is an example from Hydrilla, another invasive aquatic plant, really heavily managed in the southern tier of the United States, Atlantic and Gulf coastal plain states. Really heavily managed with fluoridone. These are dose response curves for 4 different, I'll call them accessions, 4 different populations of Hydrilla, for which they noticed they were having different amounts of control. So went to a lake and sampled Hydrilla, and this dose response curve comes from Hydrilla from that lake. And this is sort of the wild type susceptible. In Florida, they use Hydrilla around 12 to 18 parts per billion. All right. So fluoridone works on those guys. But then there's, from these other 3 populations, there's varying levels of resistance to fluoridone. And for these 2, fluoridone's off the table. It doesn't work at all on these guys. This guy, it works, but you have to use a higher concentration, and it kind of has some non-target effects that are not so great, but a manager might be willing to do it. Through a whole lot of excellent work that was not done by my lab, we know the genetic basis to the variation in these phenotypes. It's a very simple genetic control. There are 3 different amino acid substitutions at 1 amino acid position in the phytoene desaturase enzyme. That's the enzyme that's targeted by fluoridone. And so very simple mutations at 1 spot lead to these different phenotypes. And so now, right, we don't have to do dose response curves for every population of Hydrilla that we want to manage with fluoridone, right? It's very easy to just sequence a piece of DNA that has this piece in it. And my lab does that as a service for some folks in Florida. So they send me plants, I sequence that gene, tell them how many of their plants have these different DNA sequences, and then they can make their decisions from there. So that's where I hope to get with, this longer term sort of vision. And I think, we can do it with Hydrilla, we can do it with Eurasian Watermilfoil, we could do it with a lot of other different things. I think in the interest of time, I'll stop there, except just to say that there's all that sort of everything that I've shown you has really been pieced together with sort of small to medium-sized grants from a variety of different agencies and entities. And there's a whole bunch of people. They're probably not all up there, and probably most of you probably don't know who they are, so I won't list them by name. But this is really done with a lot of interaction with a lot of different people. With that, I'll say thank you, and I'll shut up at least for a second and answer any questions that you have. Thanks. Yeah, Rob.
So if this is all in the context of controlling invasive species, are you as interested in the genetic controls on the competitive fitness of the plants as the species of plants? Okay, I think you want to understand the interaction of which genes you're controlling for with your herbicide, with which genes are actually more competitive genes.
Yeah. So the question is, am I interested in genetic control of things that are not just herbicide response, and specifically how those invaders are competing with native plants? Yeah. So one of the traits that we're really interested that we have developing is what we call segregating populations for is just vegetative growth rate. So what we think happens is that these plants are just faster growers than other plants. There's a variety of ways it can happen. It could be phenological. They could come in the system earlier and then, sort of have a priority effect over other species, or they may just have faster growth rates. They may be allelopathic. They may, not be as susceptible to herbivores and any number of things. So yes. Am I interested in it? Yes. Where am I focused? I'm focused on the herbicide response because I think, relating that to those herbicide dose response curves is where, since we use those curves to determine what we're going to dose a lake, that's the phenotype that I'm most focused on. But in a general sense, yes. And, I think also thinking about it from a biocontrol perspective would also be, really helpful as well. And I suppose, there's interest in this. I just don't know what the timeline for this is. If you knew what genes were sort of making it invasive in the first place, then you might, this is where you're going with it, you might be able to have designer strategies that target those genes, to knock those down or knock those out. That's a really intriguing idea, and it would be really, really cool to do. I think there's a lot of barriers to implementing any control based on that. But in the long term, absolutely. So what we're focused on right now really is getting the methodology. See, they're non-model organisms, so they're not to whine, but they're hard to work with. And so, but it doesn't make sense to study other organisms when you're trying to manage the one that you're managing, right? So we're trying to develop these tools, and when we have those tools, and I think we're doing pretty well, we can apply those to a whole suite of traits, and hopefully the door, will really open up to do lots of things that could be cool. That's a very long answer. But.
Oh, I have a follow-up question to that.
Yeah.
So a species has to be effectively susceptible to the herbicide to become a management practice, right?
Mm-hmm.
Across a pretty wide set of areas. So why is the frequency of resistance to that herbicide so low to begin with?
So,
and is there not a fitness cost that infers? So, yeah. So to get back to his point is that if there's a fitness cost, what is the play? How do these things play out?
Yeah. So the question is, I think part of it is, well, why is resistance at such a low frequency? And then are there costs to that or not? So empirically, I don't have answers for that here. So I would argue we don't know, because we've just started incorporating genetic, this sort of, the way it's always gone for Eurasian watermilfoil has been this. I mean, people who probably would be maybe upset with me for saying this, but this is the way that I see it. We apply the herbicide, we go out 2 weeks later, and we're like, yep, good job, right? And so often the person that's contracted to apply the herbicide is the person evaluating the herbicide treatment, right? There's a big conflict of interest. So I would argue, and when we when we have expectations for how much control there should have been, and there's a deviation from that, the list of explanations has historically been things that are like, well, maybe we didn't achieve the right target concentration. Maybe there were too many boats in the water that day, or the ducks were, flapping around, or whatever. And those are somewhat reasonable explanations, but I'm very long-winded, you know this about me. Short answer is, we don't have a handle on how common it is at all. I would argue, just from those 10 sort of haphazardly sampled genotypes, looking at 2,4-D, I would argue that resistance to 2,4-D might be pretty common, but we don't have any data on it. If it's not common, it may in fact be because there's a cost to being resistant. And so if you use the herbicide and then you rotate off the herbicide, maybe those genotypes get outcompeted. In this system, we don't have any knowledge of that. And one of the things I hope happens from this, if nothing else, is that if people get excited about this and incorporate this, at least we'll be able to learn some answers to that. And we might find out in the end that, yeah, there's some resistance out there, and but it's probably more effective, cost effective to just ignore it because it's so infrequent than it is to get all up in arms about it. And I'm biased, but I'm not convinced that that's the case. I think it's probably much more common than we think in these systems. Does that answer your question, or at least touches on it? Yes, sir.
Is milfoil similar enough to any other aquatic plants that when you use 2,4-D or fluorine in an ecosystem, you start killing other plants other than?
Yeah. So the question is are there other community members that are susceptible to these? Yes. And so in the beginning of the talk, when I talked about how these natural systems are complicated from a weed management perspective, that's one of the big issues. Is how do you selectively go in and get rid of one thing and leave as much of the other things as possible? So yeah, there are lots of impacts on other plant species, and something that a permit or a regulator, or a state agency person has to really wrestle with is, well, how much are we willing to to lose for that? Back to Roger's, if we had a really designer sort of drugs, right, then we could really be more selective with those. So the way that it works typically. So watermilfoil is a dicotyledonous plant. And so most of the herbicides that are used on watermilfoil are monocotyledonous plants, have an inherent tolerance to those herbicides. And it just so turns out that most aquatic plants, it's not really true, but most aquatic plants are, many aquatic plants are monocots. And so, you know, so it will kill off some other dicots. What traditional aquatic plant weed science does is tries to find the sweet spot. How much of an herbicide can you use that has the most effect on the target species and the least effect on everything else? And then you get into a lot of philosophical stuff there. But certainly northern watermilfoil is very similar to Eurasian watermilfoil. And there are these stories that northern water, you know, we don't kill northern watermilfoil, we kill Eurasian watermilfoil. And as far as what we've seen in, you know, I don't have data to show you for that, but you take my word for that. I mean, that tends to be not true. Northern watermilfoil tends to be killed when you apply herbicides to kill Eurasian watermilfoil. So there's almost this kind of, I don't think anybody really cares from a conservation perspective about northern watermilfoil all that much. Again, that's just my sense. But you know, in areas that have heavy Eurasian watermilfoil management activities, you really can't find northern watermilfoil. And so it's almost like kind of, I get the sense it's kind of being driven extinct, you know, both by competition from Eurasian watermilfoil, but also in the efforts to kill Eurasian watermilfoil. Yes, sir.
So is there somebody else there? Yep. So what about spraying across time? If you pointed out that if you spray the same thing every 3 years, maybe, that'll select for the resistant ones. Now, so a person might do that, or he might spray different things. He might talk to a different distributor each year, and he'd choose the one this year, the next year. And I'm sure you've thought through what would happen if you use those different strategies and how you might use one or the other to your advantage.
Yeah, so the question is about basically using herbicide rotations so that, you know, if you are selecting with one herbicide, then you're using a different herbicide next. And so that genotype is unlikely to be resistant to both of those herbicides. So that's a real common resistance, what we call a resistance management strategy in weed control, certainly really common in terrestrial agriculture settings. I won't get into the gory details about it. That's something that's promoted in aquatic plant management, but at every given lake, it's actually pretty difficult to implement. There's very few options to begin with. And then there's, at the beginning I mentioned there's a lot of stakeholders, and these are multi-use systems. So you have a lot of competing, when you have a lot of stakeholders at the table, it's great, but it's also annoying in that there's a lot of competing interests, so there's the potential for competing interests. And so herbicide rotations and herbicide mixtures are promoted as a resistant management strategy in aquatic systems. As a practical matter, they're almost never used. So, and a lot of that's due to cost, and a lot of it is due to the permit issuer not being comfortable with how selective and efficacious something is going to be. The industry would love to use it, right? So one of my arguments, it's a strange argument, but I actually think where we're at with aquatic plant management is it's better to be reactive than proactive. Not because I typically think that in my worldview for most things, but as a practical matter, I think the best we can do right now is just monitor and measure whether or not an herbicide is working. And then just be honest about it. And if we decide, no, it's not working, that should be compelling argument to make a switch. And that should be compelling to the user, to the manager, and to the regulator. But in the absence of any empirical information to compel people to change, the current state of things is that they really don't change. I don't know if that touches on you. I'll go over here first.
Yeah. So in thinking through this, a lot of questions arise to me kind of coming from optimization, Bayesian decision making background. Have started thinking about, well, suppose we had this library and we knew all the genotypes and how susceptible each one was to all these different herbicides. It wouldn't necessarily be the right strategy to go out and collect 100 plants from every lake and get their genotypes and then come up with targeted strategies for each lake. I mean, that would be an extremely expensive way to approach it. Have you thought about kind of how do you approach this from kind of a value of information, cost of information? You might say there's some lakes that before we even go there, we pretty much know what's out there. We just are going to go ahead and treat them without any sampling. Others, we have, have people started thinking about that?
Maybe. I've thought about it a little bit, but from a very, from in my limited sort of capacity to it. So you bring up so the question, I'm supposed to repeat the questions, I think. The question is, are people thinking about it from an optimization perspective of like, maybe it doesn't make sense to collect a lot of information on this lake, but maybe it makes sense to do it over here. And how do you make the decision to do it or not? People do, I mean, when I talk to people about this, like when I'm sort of soliciting lakes to like work with, I'll have that conversation with them. I'll say, well, you have to think about whether or not this makes sense or not. Here's my pitch of what the information it might give you. Here's the information, the broader information that it might contribute to. And if it's not a one that works for you, it doesn't work for you. So I'd love to talk more about that with you, because I think ultimately that's where it needs to go. But
turn that into a formal decision problem.
Yeah.
Roll in, what is the cost of getting the treatment wrong? What is the cost of collecting information?
Yep.
Those kinds of things. Trading, how do all those things trade off?
Yep. And my sense is the payers, the people who pay for all this stuff, right? Because that's usually not the state agency that's regulating it. It's usually sort of private money in one way or another. That they don't have formal algorithms for that, but that they're really thinking about what the value of it is. And I do think that they kind of think through that. And so some of them, I think, if you're treating like a private pond or something like that, I mean, that's a very different, the value that you stand to get from doing something like what I'm talking about is very different than if it's a large public water body with multiple uses. And, you can think more short term in like a private pond and less about non-target impacts than if you're on a large public water body that you're thinking like, okay, this is a management program and it's going to go for perpetuity. And how do we, and we're strapped for cash and we're always going to be strapped for cash. And, so how do we get the biggest bang for our buck? So I think people think about it like somewhat intuitively, but I think it would be much better. So we should talk, and, that would be a great decision-making tool for them, because they're always limited by funds. And what they tend to put their money into is like putting herbicide in the water. And at some rate, it seems to me that's a bad decision, right? But it always seems better to sort of put your money into like actually doing something than like, I don't know, getting 23andMe done on, 100 plants or something.
Okay, we're gonna have to stop there. Thanks.
Thank you very much. I appreciate the opportunity.